Hello, everyone. Today we're going to talk about Molnupiravir, which is the oral COVID-19 drug from Merck that has recently been approved by the NHS in Great Britain and is they've submitted for emergency use authorization with the FDA, and the EU is also looking at it. That's based on the strength of the interim analysis of their phase 3 move out trial, which they had a press release, so we don't have the full data, but we will have access to the full data once the EUA gets fully submitted. But basically, this shows that they did a study in an outpatient setting with molnupiravir, and it reduced the risk of hospitalization death by 50%. These are well-designed phase 3 trials. Whenever a pharma company does a phase 3 trial, it's very well designed and they have good numbers. So usually the design is good and these are reliable numbers. What's important about molnupiravir is that it's an oral drug. And so it's very easy to provide early in the outpatient setting. The problem with remdesivir, which was the other antiviral that was looked at earlier, is that that's an IV formulation, has to be injected, and that's a little bit more difficult to deliver early on in trials. So I know you guys have probably already seen videos or news stories about this part of the thing. So in this video, I just wanted to go over what is molnupiravir, how does it work, what's the mechanism, and some of the potential pitfalls of the medication. So this is a Nature article called Molnupiravir Coding for Catastrophe. And that basically, the title sounds bad, but basically what it's saying is molnupiravir, the way that it works is it causes catastrophic mutations in the viral genome and makes the virus non-functional. And it's called lethal mutagenesis. Here's the way it does that. And in order to understand a little bit, you have to understand some basic molecular biology. So what happens during our RNA replication is there are four bases that RNA is made out of. That is A, U, G, and C. So adenosine, guanine, cytosine, and uracil. And they pair up with each other. So when it's synthesizing, it reads this line and it inserts the corresponding matching base pair. So A in the DNA strand matches up with U. If it was DNA to DNA replication, A would match up with T. So A and T match up, and in RNA, instead of T, thymidine, it's uracil. So it's AU. And then G's line up with C's, cytosine. The way molnupiravir works is that it competes very well with C. So instead of putting C, it's like when it sees a G, it should put a C next to it. But instead of putting the C in, the RNA polymerase will put the molnupiravir, MTP. Molnupiravir gets converted to its active form once you ingest it. But anyway, molnupiravir gets substituted into C. And then what's interesting about molnupiravir is that after they make it, there is an enzyme that comes through and proofreads it and makes sure that the right base pair was placed. And if the wrong base pair was placed, it either cuts it out and fixes it, or it scraps the whole thing and makes it restart. And usually it corrects it. What's interesting about this is that the proofreading enzyme does not recognize that an error has been made. It doesn't recognize that the C has been replaced with molnupiravir, and it just keeps making the strand. And so every time there's a G on the template, there's a 50-50 chance, roughly, that it'll substitute the molnupiravir instead of cytosine. And then what's interesting about it is molnupiravir sometimes looks like a uracil, and sometimes it looks like a C. So then because when it reads it, the body or the polymerases don't know what it's reading, it makes a mutation there. So then you have a bunch of point mutations, probably hundreds in there. And with that many mutations, it's lethal mutagenesis, and the virus becomes non-functional. So that's great. And that's the way it works. There's several other drugs um, like favipiravir that work the same way. Remdesivir works a little differently. It's also a analog nucleoside, but it works a little differently because it causes the viral replication to stop. It doesn't continue. Um, but here's the issue with molnupiravir, and that was talked about in this study where it says BDN4 hydroxycytosine. This is actually what molnupiravir is. So molnupiravir, this is the initial metabolite of molnupiravir. It becomes converted into this. This is the active form. And basically what they did is they looked at this in hamster cells in vitro, and what they showed that this actually causes DNA mutations in these hamster cells. And the way it works is, so RNA stands for ribonucleic acid and DNA stands for deoxyribonucleic acid. They're very similar. This is the sugar backbone of the molecule. So this like backbone, this little blue line here, this one is formed with a bunch of these, and the RNA is formed with a bunch of these. Now you see that there's only one difference between these two, and the only difference is that in ribose this is a hydroxy group here, and in DNA this is a hydrogen group. So there is an enzyme that can convert between these two. And if you take molnupiravir, which looks like this, and yeah, so here's molnupiravir right here. It gets converted to N4-hydroxycytosine, and you see it looks very similar to this. Okay, this is kind of rotated. So this part right here is this part right here in this picture, and this part right here is this part right here. So if you look at this, the only difference between cytosine and hydroxycytosine is that this amino group here is has a hydroxy attached to it. Instead of two hydrogens coming off this nitrogen, it has one hydrogen and one hydroxyl group coming off of it. So that's why it's very similar, and that's why the RNA polymerase confuses these two and inserts this instead of this. But this hydroxy group makes it so that it can be read as a different nucleoside. It can be recognized as either a C or a U by the RNA polymerase. Okay, so now what happens is if this gets converted to a deoxy, basically the DNA form of molnupiravir, now this thing can be inserted into the DNA and not the RNA, and it can cause mutations. And that's what they've noticed in this study. And what we know is that favipiravir is actually teratogenic, it's mutagenic as well. So they know that other drugs that work in this manner also cause DNA mutations. So there is a possibility for molnupiravir to cause DNA mutations, and that is like there's a mechanistic reason on how it can happen, and there is experimental proof that shows that it happens. Now, we are always exposed to mutagens. Like sunlight is basically a mutagen and causes mutation, but this is a pretty strong mutagen. And the FDA requires a lot of tests, such as the PIG-A assay and the positive, like a Ames test, and the Big Blue assay. These are tests of genotoxicity and mutagenesis, and drugs need to undergo this testing before it's tested in people. So the fact that Merck went into phase 3 trials with molnupiravir means that they passed these studies, which means that the mutagenicity is not high enough to trigger a toxicity a stoppage. So that's good, but the fact that it does cause mutations in vitro and that there's a mechanistic reason by which it could do so is concerning, and they've they've approved other drugs similar to this, like the favipiravir, but with restrictions. That means like they wouldn't give it to pregnant women because it would cause birth defects, and probably a couple other restrictions. So these are known to be, like favipiravir is known to be teratogenic, and most likely molnupiravir is also teratogenic and causes mutations. To what extent is unknown, but DNA mutations can lead to cancer is one of the biggest things, or birth defects is probably the second thing. And some of those things are not, they won't be picked up in early trials. It may be a long-term effect. So while molnupiravir seems to be effective clinically against COVID-19, there are some safety concerns with this. And so people should be aware of the safety concerns of mutagenesis with this. It is going to be a oral form drug for the early treatment of COVID. Will be a game changer. And since COVID is endemic now, we are probably going to need, as I've said before, therapy is going to be probably a more important weapon than eradication. So I will link these studies below in the description if you guys want to look at them more carefully. And maybe in a future video, I'll talk about how remdesivir got a lot of bad press early on, mainly because it showed ineffectiveness in the in-hospital setting. There are new studies showing that it may be more effective in the early setting, and they are working on an oral form. So if they make an oral form and do a phase 3 trial in the early trials, that'll be very interesting because remdesivir has a much less mutagenic potential.